This next mechanism of introducing different genes to bacteria is transduction. And transduction is the introduction of genetic material by way of a bacteriophage. And a bacteriophage is a virus that is specific for bacteria. And if you take a look at that bottom picture, there is a picture of a bacteriophage. To me, it looks very robotic, almost alien-like. It reminds me of those little hex toys that you might see at Target. So it looks almost like out of this world and almost mechanical. That is a bacteriophage. A bacteriophage would not affect our cells. It would only affect bacteria. And if you remember talking about the gram-positive bacteria, and we talked about that teichoic acid, potentially it could be a docking station so that that bacteriophage can actually attach to that cell. If viruses can't attach to the cell, they're not going to be able to replicate. So in order to really talk about transduction, we're going to have to talk about a little bit about phages as well as the viral replication cycle. Remember, viruses are not considered living things. They are not cells. They cannot undergo the characteristics of living things like metabolism, maintaining homeostasis, reproduction. In fact, we don't even say that viruses reproduce. We usually say that they replicate. They have to take over the host cell machinery in order to replicate. So they're what we would call obligate intracellular parasites. So in order to really talk about how these phages introduce genetic material, there's a couple of different types of phages. And there's a virulent phage. This is going to take over that cell machinery right away, and it's going to cause lysis of the bacteria. It's just going to kill it. A temperate phage is almost like a more modified phage, where it's not going to cause death immediately. And that phage DNA is actually going to be incorporated into that bacterial chromosome. That's when we call it a prophage. And it's just going to kind of hang out there for many, many generations. It's not going to kill that cell right away. Bacteria has no idea that it's there. So in order for these phages to kind of take over the cell and undergo replication, there's a couple of different forms of viral replication. And so I just want to introduce this a little bit. We'll be talking a little bit more about this in chapter 11 on viruses. So this is just a little introduction, a little simplified view. And these are the lytic and lysogenic cycle, or lytic cycle, however it is that you prefer to say it. So if you take a look at the left-hand side, this is a snapshot of the lytic cycle. So there you can see that bacteriophage is attaching to the cell. It's injecting its DNA, and then it's taking over the host cell machinery so that it can make its parts and then assemble the parts, and then it blows open that cell, lyses that cell, and now we've got all of these bacteriophages going out, and they can infect other cells. This is the violent, quick cycle, and that's going to take over the host cell machinery right away, and it's going to kill the cell. Lysogenic cycle is a little different. This is the longer, sneakier cycle, as I call it. So at that top picture on the right-hand side, you can see that bacteriophage attaching to the cell. It's injecting its DNA, but it doesn't take over the host cell machinery right away. You can see where it's incorporated into that bacterial chromosome. This is when we call it a prophage, and it's just going to kind of hang out there for a while, for many generations. Cell has absolutely no idea that that viral DNA is incorporated into its chromosomal DNA. So it's going to undergo many cell divisions until perhaps something happens in the environment. Maybe the cell is stressed out, it's been exposed to chemicals or radiation, and that DNA is like, whoa, we are out of here. And then it will actually cross over into the lytic cycle. Now it's going to take over the host cell machinery, make its parts, assemble its parts, and then blow open the cell. So these are two viral replication modes. And so to really understand transduction, we need a little preview of the lytic and the lysogenic cycle. So the two types of transduction that we'll talk about are generalized transduction and specialized transduction. So with generalized transduction, this is going to utilize that virulent phage. That's the violent phage, right? So this is going to prompt it to go into the lytic cycle. So this is that quick cycle where it's going to take over the host cell machinery and blow open that cell. So in this picture that I've drawn on the right, we've got our bacteriophage with the red DNA. That's the bacteriophage DNA. And it's attached to the bacteria. And you can see the blue circular chromosome. So that virulent phage is going to attach. It's going to inject its DNA. And then what you're not seeing from this picture is it's taking over the host cell machinery to make its parts, assemble the parts, and then blow open the cell. Well, that resulting bacteriophage, if you look at the products down below, some of those bacteriophage are packaged with the bacteriophage DNA. And because this is kind of a messy, chaotic cycle, you'll notice that one bacteriophage has the bacterial chromosomal DNA. So it was actually packaged with that DNA in error. So here's where the scrambling of genes comes in. These bacteriophages can now go out and they can infect other cells. So now another cell can actually have access to these genes. So looking at that bacteriophage with the blue DNA, with the bacterial DNA, what if that was actually packaged with antibiotic resistant genes? Now a bacteria potentially could have access to those antibiotic resistant genes. So a way to kind of scramble those genes. So this is a case of generalized transduction. And here's another picture where you can see that virulent phage. And this picture is a little more detailed. The DNA is injected. It's going to take over the host cell machinery. So you can see where the parts are being made. Then they're being assembled, and then they're being released. If you notice in that second picture on the right-hand side, that B bacteriophage has the bacterial DNA in it. And now, if you can see with that picture, now it's injecting that DNA into the cell. You can see where that DNA is now incorporated into the genome. Now, if that B DNA had an antibiotic-resistant gene, now that bacteria has access to it. So again, another way to introduce genes and that genetic diversity. The second type of transduction is specialized transduction. This is when a temperate phage infects the cell and it's going to undergo the lysogenic cycle. So in that picture, we've got our bacteriophage. And it's going to inject its DNA, but it's not going to take over that host cell machinery right away. This is that longer, sneakier cycle, the lysogenic cycle. That bacteriophage DNA is now going to become incorporated in the bacterial genome, and it's going to exist as a prophage. This bacteria has no idea that that DNA has been incorporated. It's going to go about its business, and we're going to go through many, many generations until something happens in the environment and then it crosses over to the lytic cycle. Now that phage DNA is going to take over the bacterial machinery, and now it's going to make its parts, assemble its parts. So because that prophage has been intermingling among that bacterial DNA, some of that DNA is going to be combined with the bacterial DNA. So if you take a look at the phage on the bottom, you can see where it's a combination of the phage and bacterial DNA. So they're almost like brand new phages. So again, incorporating those different genes. And again, here is another detailed picture where we're injecting, that phage is injecting its DNA. It's existing as a prophage, many, many generations until it crosses over to the lytic cycle. And now all of those phage that are being released now have a combination of the phage DNA and the bacterial DNA. This could have some potentially antibiotic resistant genes or other useful genes that bacteria that is now being infected with that new phage can actually incorporate. So ways by way of a virus that genetics can kind of be scrambled and increasing that genetic diversity.